Today, I'd like to welcome you all to this seminar. We're going to hear from Srini, who's going to be talking to us about predicting lodging in sorghum breeding trials using phenomic and genomic approaches. Next slide. My name is David Jordan. I'm a professor in plant breeding and genetics at the University of Queensland within the Queensland Alliance for Agriculture and Food Innovation. And I'm actually Srini's PhD, one of Srini's PhD supervisors. Heard the presentation before, and I think you're going to enjoy it. Next slide. So I'd like to take the opportunity to acknowledge the traditional owners and the custodianship of the lands on which we meet today. And on behalf of the traditional owners, I pay respect to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. Next slide. Just a few housekeeping issues. The seminar is scheduled between 12 and 1. At the conclusion of the seminar, we'll hold a Q&A session. If you have any questions that you want answered, please use the Q&A button at the bottom of the screen. Don't use the chat button because we won't be answering those questions. We won't see them. And we'll address those questions at the end of the seminar. So you can ask questions as you go along. You don't have to remember them to the end. Okay, next slide. So without further ado, I'd like to introduce Srinivasa Mothukuri. I'm not very good at my Indian pronunciation, but I'm sure Srini will give me some more lessons. Srini studied for his master's degree in plant breeding and animal breeding at the University of Göttingen in Germany. Göttingen in Germany, sorry. His master's thesis was conducted in collaboration with CIMMIT, looking at different strategies for using sparse phenotyping and genomic prediction in maize. His PhD with us at QAAFI focuses on novel approaches to improve lodging resistance in sorghum, particularly phenotyping and generating good phenotypic data. And his work will be focused on developing a UAV-based approach to measure lodging, because this is a difficult trait for breeders to measure. His project is aligned to a large GRDC funded project with partners Advantage Seeds, the Queensland Department of Primary Industries and the University of Queensland. And without further ado, I think it's time for Srini to start his presentation.
Thank you very much, David, for introducing me. Good afternoon, everyone. Thanks for joining my seminar. I too acknowledge the traditional owners and their custodianship of the lands on which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognize their valuable contributions to Australian and global society. So today I'm going to talk about my PhD research on predicting lodging in sorghum using phenomic and genomic approaches. Sorghum production in Australia is highly variable from year to year due to seasonal rainfall. Sorghum is a rain-fed crop grown on clay soils with high water holding capacity. Farmers usually plant only when enough water has accumulated in the soil profiles. If rainfall is limited later in the season, the crop runs out of water during grain filling stage. This late season stress is called terminal water stress, and terminal water stress is common and it is a major driver of lodging. Lodging means plants fall over onto the ground before harvest. Lodging is a significant challenge in sorghum as it is in other cereal crops. However, unlike many other cereal crops, sorghum can remain alive after grain filling if water is available. This makes the expression of lodging more complex. In Australian sorghum growing environments, lodging often occurs when the plant expresses terminal water stress. Terminal water stress reduces photosynthesis during grain filling stage. As a result, carbon is remobilized from the stem to the panicle to fill the developing grain. This weakens the stem, contribute to plant death, and increases the risk of lodging. Therefore, lodging is a major cause of direct yield loss for growers, but it is also has another effects that may be even more important. As I mentioned on the previous slide, lodging is a major cause of direct yield loss for growers, but it can also have important indirect effects. The direct yield loss is obvious because the harvester may not be able to pick up grain from the lodged plants. However, the indirect yield loss is more subtle. Farmers often strongly biased against hybrids that have a high risk of lodging. As a result, they may prefer safer hybrids with a lower chance of lodging, even if those hybrids have lower average yield potential. This means that potentially high yielding hybrids may be avoided because they lodge more frequently. Over time, this can reduce the average yield achieved by growers. Therefore, developing lodging resistant varieties is a top priority for both public and private sorghum breeders. Genetic improvement requires consistent trait measurement. Plant breeders need to be able to measure lodging at this scale. Plant breeders currently rely on visual scoring approach to measure lodging across thousands of plots. The problem here is measuring lodging across thousands of plots visually, which is time consuming, expensive, and may not be accurate. Therefore, a practical alternative is measuring lodging using UAV imagery, which can be high throughput, lower cost, and may increase accuracy of measuring lodging. To do this, we collected phenotypic and UAV imagery data from 3 different sites. Each site contained 2 experiments. For this study, we had 3 types of data available. For my research, we used manual counts where we counted the total number of plants and number of lodged plants in each plot and converted this into a percentage scale from 0 to 100. Manual counting is very labor intensive because it takes around 1 minute per plot to count every plant in a plot and lodged plants, and therefore it is not commonly used in practice. However, manual counting is potentially more accurate. What breeders use currently, they use visual scoring approach to measure lodging in current breeding trials, where they visually rate each plot for the degree of lodging on a scale from 1 to 9. 1 indicates 0 to 11% of lodging, and 9 indicates 88 to 100% of lodging. These visual scores are categorical values from 1 to 9. So visual scoring is much faster, taking around 15 to 20 seconds per plot. The manual counts and visual scores are positively correlated. The third data type was UAV imagery. My main aim of this research is to develop an approach to measure lodging using UAV imagery. I'm developing an approach to measure lodging using UAV imagery. To do this, I used manual counts and UAV imagery data from 2 sites. Then developed the model. Then the developed model was validated at a site where we have visual scores and UAV imagery. To develop the approach using UAV imagery, the drone with a RGB camera flew over the trail following a planned field path. During each flight, the drone captured thousands of overlapping images from different angles. Those images were imported into photogrammetric software where they were aligned and combined into a single image of field called orthomosaic. The software then converted the two-dimensional images into three-dimensional point clouds, allowing us to estimate the height of different pixels within each plot. We used UAV imagery taken at two different crop stages to analyze change in pixel height. The first set of images was captured around the flowering stage. At this stage, no lodging had occurred and crop had reached its maximum height. The second set of images was captured on the same day as the manual measurements were taken. By this time, lodging had occurred, so the change in pixel height between the two stages could be used to estimate lodging severity. Here is an example from a single plot. The image shows a two-dimensional representation of the three-dimensional point clouds. The x-axis shows the row length of the plot and y-axis represents the density of the pixels at different heights. The different colors illustrates height sliced based on selected predictors, percentiles. The height change between two different stages of the plot is due to lodging. To capture these changes, we generated multiple predictors by subtracting high height at flight 1 with the height of 95th, 85th, 75th, and 65th at flight 2 separately. Those predictors were used in various statistical models, such as multiple linear regressions and a range of parametric and non-parametric models. Parametric models assume a constant linear relationship in the data, while non-parametric models can account for non-linear relationship in the data. Each model has its own advantages and disadvantages. Therefore, I explored another strategy using an ensemble naïve average approach, which leverages the strength of all individual models by combining their predictions and averaging them. I call it naïve because it assumes that all models contribute equally without weighting any model more than the other model. To select the best performing model, we used standard cross-validation approach. The complete data set included the manual counting and 4 selected predictors from 2 sites. The data were then randomly split with 80% of the plots allocated to the training set and the remaining 20% of the plots allocated to the testing set. And I applied the statistical model shown on the previous slide to train the model. Then the manual lodging counts in the testing set were masked and lodging was predicted using the trained model together with the selected UAV derived predictors. At this stage, we had both the observed manual lodging counts and UAV predicted lodging counts for the testing set, which we used to evaluate model performance. This entire process was repeated 100 times to provide a robust evaluation of model performance. To evaluate model performance, we used 2 metrics. One is Pearson correlation and the another is residual mean square error between observed and predicted plot values. In the corresponding plot, the x-axis shows models and y-axis represents their matrix. Each block dot illustrates the accuracy from one of the 100 iterations, and the red dot shows mean across 100 iterations. We are looking for model with high correlation and low error. When comparing all the models, we observed that in general, non-parametric models outperform than parametric ones. However, our study found that ensemble naïve average model performed slightly better than the other individual parametric and non-parametric models. This is because each individual model captures different aspects of the data, and some features are well predicted by one model but poorly predicted by another model. By averaging the predictions, we are leveraging the strength of all the models, resulting in slightly better predictions than those of the individual models. Then we selected the ensemble for further analysis. After selecting the ensemble model for further analysis, we examined the observed and predicted lodging values across the 100 cross-validation iterations. We could see that some plots had much large difference between the observed and predicted values than others. So we wanted to understand why these plots had large difference between observed and predicted values. This figure included results from multiple cross-validation iterations. So a single cluster may represent repeated predictions for the same plot. The x-axis groups plots by their observed lodging percentages, while the y-axis shows the squared residuals between the manual lodging counts and UAV predicted counts. Most plots had relatively small prediction error, but some plots showed very large difference between observed and predicted lodging values. These plots appeared to be potentially outliers. Therefore, we developed a criteria to identify these plots for further visual inspection. Using this criteria, we identified 180 potential outlier plots out of 2,675 plots. We had photos of every individual plot, so we compared the plot images with the recorded manual lodging counts and the UAV predicted lodging counts. We observed two main issues from this visual inspection. First, this is an example of a consistent prediction. The manual lodging count was 90%, and we can clearly see a high level of lodging in the plot image, that plot image we got from the UAVs. However, in some plots, the manual and UAV value were highly inconsistent. For example, in this plot, the image shows no lodging with the plant still standing upright. However, recorded manual lodging count was 90%, while the UAV predicted lodging value was 0%. From the image, it is very unlikely that this plot would have been counted as 90% lodging in the field. Therefore, this inconsistency was more likely due to a transcription or plot assignment error, where the wrong value may have been recorded or assigned to the wrong plot. Overall, we identified 82 out of 2,675 plots appeared to be affected by this type of transcription or plot assignment error. The second issue involved plots with poor establishment. In some plots, establishment was very poor, resulting in fewer plants and more visible ground. When we examine the plots with high residual, high prediction errors, we found that many of them had poor establishment. In practice, breeders would usually exclude poorly established plots from phenotypic analysis because poor establishment can compromise the reliability of lodging assessments. So the two main sources associated with high prediction error were like transcription error and also poor establishment. After identifying these two issues, we wanted to know whether removing these problematic plots would improve model performance. Here I am looking at the impact of transcription errors and also poor establishment on model performance. The reason for doing this is that if the model is trained using problematic plots, those errors may influence the model and affect its predictions. The x-axis shows the approaches, which you will see in the further slides, and y-axis represents the residual mean square error. Each block dot shows the residual mean square error from one of the 100 iterations. Approach 1 is the versional model, which includes all plots, including those affected by transcription errors and poor establishment. When we removed the plots affected by transcription errors from the versional model evaluation, the residual mean square error decreased from around 18% to around 15%. This shows that those plots were contributing substitutionally to the prediction error. Next, we looked at the effect of poor establishment by removing those plots from the versional model evaluation. We then calculated the residual mean square error again. When those poorly established plots were removed, the residual mean square error decreased only slightly. This shows that poor establishment was contributing to prediction error, but it affects was smaller than the effect of the transcription errors. After that, we removed both known sources of error, the plots affected by transcription and the plots with poor establishment. As expected, the residual mean square error decreased substitutionally compared with the versional model evaluation. Up to this point, we had not changed the trained model itself. We were mainly quantifying how much of the prediction error was associated with these two known problems. But the more interesting question was whether these problematic plots were actually affecting the model training. If the model is trained using plots with known errors, these observations could potentially influence the fitted model and affect its predictions. In the final approach, we removed the plots affected by transcription errors and poor establishment. Then we completely retrained the model. The most important comparison here is between approach 4 and approach 5. In approach 4, we removed those problematic plots only when evaluating the prediction error, but the model itself was still the versionally trained model with all the data we had. In approach 5, we removed the same problematic plots from the data set and we retrained the model from the beginning. As you can see, the residual mean square error was very similar between approach 4 and approach 5. This tells us that including those problematic plots in the versional training data had very little effect on the fitted model itself. So these plots increased the prediction error for individual observations. They had only a small influence on the model training. Overall, this suggests that ensemble model was relatively robust even when the training data contained some transcription errors and plots with poor establishment. We wanted to examine the predictions to determine whether site and germplasm set affected prediction accuracy. As I mentioned earlier in my presentation, we had two different sites, and each site contained two trial series. I said two experiments, those are two different trials. Those two trial series contain different germplasm. So one trial series might potentially be easier or more difficult to predict than the other. Therefore, we tested whether the model performed consistently across sites and trial series. The results showed no major differences, suggesting that prediction accuracy was generally stable across both site and trial series. We developed the model using all the data, available data, but the lodging value were not evenly distributed across the different lodging percentage groups. We therefore wondered whether this uneven distribution was affecting prediction accuracy. To test this, we compared two different training set sampling strategies. One is stratified sampling, another one is random sampling. In the stratified sampling approach, we selected a more even number of plots from the each lodging percentage groups for the training set, whereas in the random sampling approach, plots were allocated to the training set randomly. So what we found, stratified sampling provided only a small improvement compared with the random sampling, suggesting that prediction accuracy was generally stable across two sampling approaches. So far, we have been looking at how well we can predict lodging in individual field plots using UAV imagery. These plots came from design experiments that included a range of factors such as sites, genotypes, and replicates, and different germplasm groups. We can analyze these data statistically to understand how the lodging phenotypes type measured at the plot level can be used for breeding decisions. At that plot level, the results show reasonable prediction accuracy, uh, although the observed and predicted values were not identical. However, plant breeders are mainly interested in genotype ranking, identifying which genotypes are best and which genotypes are worst. Because manual and UAV phenotyping may rank genotypes differently, we also need to know and evaluate how reliable the UAV based method would be for selection decisions by comparing their relative accuracies. To do this, I used linear mixed model approach. We used a linear mixed model approach because the breeding trials data were unbalanced with not all genotypes and traits evaluated in every environment. Linear mixed models allow us to account for both the genetic structure and experimental design of the trials. This provides a more appropriate basis for comparing the relative accuracy of manual and UAV-derived lodging phenotypes. We then used two complementary analyses to estimate these relative accuracies. First, we conducted a univariate analysis where the two traits were analyzed completely separately. This allowed us to evaluate the relative accuracies of the two traits independently. We then conducted a multivariate analysis where the two traits were analyzed together. We used both approaches because the multivariate analysis can potentially borrow information between traits, whereas the univariate analysis evaluates each trait independently. However, the multivariate analysis is more appropriate for estimating the genetic genotype relationship between two traits. We found that both approaches gave consistent outcomes. Therefore, in the following slides, I will focus on multivariate analysis and their results. Here is general form of the model and components we used for the multivariate analysis. The main purpose of this model was to estimate the genetic variance, covariance, and genetic correlations between the two traits within each site and across. The genetic correlations gives us an indication of how similarly the two different measurement approaches rank the genotypes. So a high genetic correlation means that the manual and UAV based approaches are generally ranking the genotypes in a similar way. I'm going to present the results for trial series 1, and the results for the other trial series were very similar. One of the things that plant breeders are interested in is heritability, which tells us how much of the observed variation is associated with genetic differences among genotypes. The heritability estimates from the two phenotypic methods were very similar within each site. Here I am comparing between methods within the site. This suggests that the UAV-based predictions captured a comparable proportion of genetic variance to the conventional phenotypic method. However, similar heritability alone doesn't tell us whether the two methods rank genotypes in the same way. Therefore, we also looked at genetic correlations between the two phenotypic methods within each site. So the high genetic correlations within the same site indicate that the genotype rankings from the two methods are very similar. This is what we expected because these two sites we used for model development. However, we also want to know whether the model has broad applicability behind the sites we used for model development. So now we have the validation site. As I mentioned in my early slides that we developed model on two sites and we validated on a site where we have the visual scores. Where we have the breeders visual lodging scores and UAV lodging counts. These visual lodging scores are the ones I mentioned earlier where the breeder rates each plot using 1 to 9 categorical scale. Importantly, this was a completely independent site and year, and it was not used for model development. At this site, the overall heritability was lower than the model development sites. However, the visual scores and UAV lodging counts showed similar and consistent heritabilities together with the moderate genetic correlations between the two methods within that site. So even at the validation site, the UAV based approach captured a similar level of genetic information to the breeder's visual scoring approach. Breeders use selection differential to estimate the effect of selection. It is very simple concept. It is the difference between the mean of the overall population and the mean of selected genotypes. In our study, we selected top 10% of most lodging resistant genotypes using each phenotypic method within each site. To compare the two methods fairly, we express the results on the same lodging scale. These numbers shows that the expected reduction in lodging when we select the top 10% of genotypes with lowest lodging and use them for the next generation. For example, at Jandavi, selections based on manual counts reduction lodging by about 13 percentage points, while selection based on UAV predictions reduced it by about 11 percentage points. Overall, the two phenotypic methods produced comparable selection differentials within each site. From breeder's point of view, this suggests that either phenotypic method could be used to make similar selection decisions. Even at the validation site, selection differentials were comparable. This is providing confidence in the utility of the UAV-based approach for current plant breeding programs. As I mentioned in the previous slide that from breeder's point of view, these two phenotypic methods are equivalent. But for plant breeders, it is not just about accuracy, it is about how much it costs to run the experiments. For example, the validation site included about 2,000 plots. For visual scoring, we included to measure those 2000 plots, we included the cost for traveling to the site and the labor required to score those plots. And we assumed that visual scoring took about 15 to 20 seconds, as I mentioned in my earlier slides. To measure these 2000 plots, it cost $1,200 approximately. Per plot, it cost 60 cents. For the UAV approach, to measure the overall cost, we included the travel cost to the site and labor required to fly the drone over the trail. We did not include the image processing cost because the processing pipeline has been automated. Using the UAV-based approach, the cost was approximately 6 cents per plot, and also to measure those 2000 plots, it cost $120. Therefore, the UAV-based approach was approximately 10 times cheaper than the visual scoring approach. These cost savings could allow breeders to phenotype large trials or more locations using same budget. This could increase selection intensity and ultimately contribute to increase the selection genetic gain. So what I am currently working on, developing large calibration sets across many sites and testers is expensive. And as I mentioned earlier, cost is very important for breeders. So we want to understand how much calibration information we actually need, particularly how many sites are required and whether different testers are important. I am currently testing whether a low cost calibration set based on just one site and one tester combined with the genomic relationship matrix can be used to predict the average lodging performance of genotypes across multiple environments. We are also uh, investigating whether we can improve the genomic prediction of final lodging by including correlated traits such as stay green, flowering time, plant height, and early lodging. Ultimately, the main aim of the what we are currently working on is to determine whether we can achieve useful genomic prediction of lodging using less phenotyping information with well calibration set. Therefore, we can reduce the phenotyping cost. The key message is that UAV-based lodging estimates have utility as a phenotypic tool. They provide comparable information to the conventional method, but at much lower cost. I'm thankful to my PhD supervisors for their support, guidance, and valuable inputs throughout my PhD research. I'm also thankful to contributors Sean, Ken, Andreas, and Misha. I'm also very thankful to reviewers for their help, helpful feedback during my progress, and also thankful. So I'm working on pre-existing data that collected by Sorghum breeding team from DPA and UQ at Hermitage Research Facility, Warwick. So I'm very thankful for them who collected the data. And I'm also thankful to ARC, Center of Excellence for Plant Success, IRTG, and also GRDC and Advanta. I would be happy to discuss any questions or comments on the work I have presented. Now I am facilitating to David Jordan for the question and answer session. Thank you.
Okay, thanks, Srini. That was excellent. And you've got some really good questions in the chat. So I think we'll start off with those. The first question, obviously from a sorghum person, is it possible to differentiate between root lodging and stalk lodging with this UAV-based method?
In sorghum, currently in Australia, we are facing stalk lodging. We are trying to measure lodging. doesn't matter for us whether it's top lodging or root lodging.
Yep.
Yeah.
So you're saying that the method would work well
Both,
for both
both.
types of lodging, but you can't differentiate, I think is
Yeah.
what you're saying.
Yeah.
Okay. And now this looks like a good physiological question. In sorghum, lodging is often associated with high grain yield. Therefore, when selecting for lodging resistance, there is a concern that we may inadvertently select against high-yielding genotypes. What are your thoughts on this relationship? What would you recommend is the best breeding strategy to improve lodging resistance without compromising yield?
So we already knew that high yielding hybrids have more propensity to lodge, and that is the reason we are trying to develop lodging resistant varieties. And but what Australian sorghum growers do, select select safer hybrids that cannot lodge, even they are fine by having the lower average yield. So that's what we are trying to do, developing lodging resistant varieties that can provide better uh, yield, better average yield.
Okay. I think the question's more about what strategy might you recommend to use this data on lodging and data on yield to select for both traits simultaneously. And that's probably beyond the scope of your current PhD, but if you have a thought, you're welcome to suggest
Um,
or not.
no,
Okay.
I don't have means.
Okay. So the next question is from Emily, who is interested in the money. So what is the payoff time for the UAV approach? And I think by this question, she's asking how long would it take to pay for the cost of, the drone and
So so we have automated phenotype processing approach. So we used only the person who can travel, the technician, or
Mhm.
uh, traveling cost and flying cost.
Yeah,
Because we have all the equipments, drone and processing
yeah.
uh, this.
So you
So
probably need to answer this question, you need to know how much a drone's going to cost and depreciation. So
yeah,
something for you to think about for later.
yeah.
Right, here's another good question, which I'm sure is from a physiologist. Were the data adjusted for height and maturity before analysis?
Uh, I think no, we didn't adjust the lodging data for height or maturity before the analysis. We looked at whether there are some potential outliers before analyzing. Then yeah, we
Yeah.
have conducted the analysis.
And when you think about your measurement, your metrics for,
Yes. So when we were generating the predictors, we normalized our difference between two different stages of the crop.
so you're saying you adjusted for the height in the metric.
Yes,
Yeah.
yes, yeah.
Okay. And the final question is from Siva, who used to work in our lab. Good talk, Srini. Could this approach be applied to other crops other than cereals where there is no head and panicle?
Yeah, thank you, Shiva. It's a good question. Uh, it's so here we took the height as the major factor. If those plants, those crops have that height factor, if we can differentiate that height, then we can measure.
So you're
So
measuring height of the canopy, not or height of the
mm,
whole plot, not the height of the individual plant. So
yes,
I guess the answer is probably yes.
yeah, yeah.
Okay, the next one's from Chang. You mentioned that you will work on staggering and lodging. How are you planning to measure staggering, and will it be assessed at one final time point or through repeated weekly measurements?
So, thanks for your question. I mentioned that we would like to use the staggering as secondary trait to improve the genomic prediction of lodging. Um, we are not assessing the staggering using UAVs.
Mhm.
Yeah.
So
So,
it's historical data.
yeah,
So it's only, I took the data, so it's only a
yeah,
single time point that we've got,
yeah,
pretty consistent across sites.
yeah.
Okay. So I'll finish up with one question.
Yes,
We
too.
found that removing the plots that had data issues did not change the quality of the prediction model much. Can you explain why that might have been? What are the potential reasons for that?
Um, it's so what we have done, so that could like it tells that the amount of the uh, proper data we gave to train the model that already uh, yeah large enough, big enough, so it used that remaining data except that 80 plots and trained properly, and it didn't show any effect on the training. And like that data set was like the wrong data was 3% in overall data set. So those models used the remaining 95 or 97 percentage of data to avoid these errors.
Okay. Well, I think we've exhausted the questions now. So if you move on to the next slide, Srini. So we should have moved on to this earlier. Next slide again.
I'm sorry. Yeah.
Yeah. Okay, so I'd like to thank everyone for joining us today, and thank Srini again for the excellent presentation. If you haven't already and you like the content here, please sign up to our seminar invitation list using the QR code here, or going to Coffee Science seminars, or that will be great. I know there's a range of interesting topics that come up every month or week or whatever it is, quite often. So that's great. So I'd like to thank everyone for turning up and for the excellent questions.